All right. Okay. I think we're about enough people are here. All right. So, if you're in this class, you should have gotten your read count data, which is basically a matrixed count or a matrixed version of our RSIM output. So basically, I pulled out the read count column from all of those files and just made a matrix of it. Right, so you'll see your sample name at the top and the gene name as a row name, right? And so from that kind of data format, we can move forward with estimating differential gene expression or differentially expressed genes. Sometimes in the literature they get an acronym of DEGs, differentially expressed genes. So there's more than one way to do this, right? I think DESeq, sometimes I know a trick or I know a tool because that's the way I learned how to do it, not necessarily because it's the best way. And every tool varies under different conditions, right? Every tool is the best tool under a different condition. Whatever. There are other R packages that can do differential gene expression. I think EdgeR is another popular one. But yeah, I just learned how to use DESeq. I think it's pretty polite. And it's consistent. It works well, and the output data is easy to plot with. So that's just my traditional tool of estimating and identifying differentially expressed genes. Now, this is kind of it's tricky to read the manual, right? The actual DESeq manual is not exactly a fun thing to learn and to read through. And honestly, so I did this last year. I did this same course in the spring last year. And so there is another video in the other semester, like the spring 2020 semester, it's effectively the exact same video. And I think that's by far my most popular video. So it suggests that DESeq isn't exactly a friendly package, but it's not all that bad to use either. Although tutorials help, nonetheless. So this video is basically going to be the same as that one. I just don't have a mohawk in that one. Right? It was kind of close. So by that time we're like 2 months into COVID quarantine, went a little quarantine crazy and made a mohawk before the semester was over. Whatever. Let's go ahead and just go through the process of DESeq and what it requires and how to run it. So again, this is all going to be in R and RStudio. You should have gotten some instructions to download specific dependencies, specifically the DESeq2 package, and a couple others. So first off, going to load in, make sure I'm starting from scratch. Library DESeq2. Okay, first thing I need. A little bit later in this script, I'll need another package, the 8GLM. 8GLM. We'll need that in a little bit. But first, what we have to do is load in our data, right? That's kind of the first step is to load in our count matrix. So load count matrix. And so this is a CSV file. Even though it says .out on it, it is a CSV file. And I guess I should change my session. I should change that session, my working directory. So I'm going to choose directory, and I have this hanging out on my desktop in a temporary folder that I've named, not that, DESeq, so I know what I'm looking for. All right, so I'm going to load in my data. So this is a CSV file, so I can just do read.csv, and then the allcounts.out, right? That's the file that we actually need, is the allcounts.out. I have to specify a couple other things in here. This just is kind of a requirement of DESeq. I have to let us know that there's a header present, like the columns have names. And my row names, I need to set my row names as well. And so my row names are going to be the G names, and that's going to be row names equals 1. So it's the first row, right? And so I think we can kind of see this data briefly. Let's see. just so we can see what's actually happening here. Okay. All right, CSV file, column names, gene names. Whoop, don't touch it. And then the recounts for each library that we analyzed, depending who analyzed it. And now, if we actually look at the recounts, most of the time, recounts are going to be pretty similar to each other across rows. Not always. That's what we're looking for. We're looking for differences across our rows that specifically correlate with the treatment, right? So we have the water treatment, and we have a PSU treatment, a saltwater treatment. Water is effectively the control, and PSU is salty water. But right, so like if a gene is lowly expressed in one tissue, it's probably lowly expressed in all. And if it's highly expressed in one, it's likely highly expressed in all, right? Usually recounts, most of the time, not always, hover within an order of magnitude of each other. And it's kind of nice. The thing about RNA-seq is that it's repeatable. It's very, very repeatable. But nonetheless, we need to load that in. Make sure our row names are classified or identified, and just load that matrix in. Wrong button. Enter. Okay. Right. So you can see that again. We have 13,000 rows, so we have about 13,000 genes present. That's about expected. About 13,000. 6 variables and blah. Right. That's our data. Now we have to give DESeq another file, or at least it's easy to give DESeq another file that gives additional properties about this column information. And so that's going to be, I've named it col data, but that's going to look something like this. Right, this is the column data. So it has our it ultimately has 2 columns. So the first column, or sorry, the first row is going to be a header or identifier. Now I don't really have to leave anything present for the first column name. And so our first column is going to be our sample names. And so recognize whenever you're making this file, this column data file, the sample name, the sample order has to be in the same order that's present in the column names of the matrix, right? So the first one is NWINS 15 PSU, then LaMaha 15 PSU, CTOVA 15 PSU, right? So these column names have to be in order, and then we apply additional columns, right? So there can be more than one column, but it's just in our specific data set, we only have one treatment. It's just going to be the condition, because they're all from the same tissue, so we don't need to necessarily specify tissue or species or anything like that. We're just specifying the condition. And it's whatever you want. It's arbitrary. Whatever helps you remember what this is. In my case, I just did 15 PSU or water. But DESeq needs this. It needs to know the sample name, whatever's in the column names, and the treatment. Right? I'm just using condition as an arbitrary word. It can be treatment, column, condition, it doesn't matter. Totally arbitrary, just keep track of it. But nonetheless, it's tab delimited, so recognize it is all tab delimited. Right? These aren't spaces, it is a tab. And we need this file to be loaded in simultaneously as our matrix data. So I have to load that in as well. So I'm just going to call that like info. And that's, since it's tab delimited, I'm not going to use read CSV. I'm going to use read.table. And so that's col data. Header is equal to true. And just for clarification, sep is equal to tab. Tab. Okay. So we load that in. Here's our info. Load it in. Same thing, we just have this X column of sample number, and then the condition column that has our treatment, 15 PSU or water. Right, so that's how your data should, that's how your column data should look like whenever you actually load this in. So data should look like a big old matrix, and this is, we don't have to specify row names. This will be inherent. We don't have to specify row names. We just load it in as in a traditional read.table fashion. Okay. Make sure my libraries are actually loaded in. I've gone ahead and applied like the link to the tutorial, or like yeah, I guess it's a tutorial made by the people who made DESeq. It took a while. Okay. So feel free to look at that. All I'm doing is more or less sampling the important parts from that given tutorial. It's kind of a pain to read. It's a little bit of a pain to read. All right, so I have my data in. Now I need to link these two files to DESeq. I have to tell DESeq to specifically make a specific data structure that's going to take into consideration these two files, and also the experiment type, what's going to be basically the test variable. So in that case, I'm going to use DDS as my variable name. But either way, the package or the function that does this is DESeq dataset from matrix. There's more than one way to do this. It just depends on what your data is, right? In my case, I've loaded it in as a single file matrix. But you certainly can. There are other functions that will let you read in, 6 RSIM files and extract the read count data. Fine. It depends. It's whatever. But read more or less the same. The variables are going to be the same whether we import from matrix or import from TX import or something. It's just going to be the data frame, or sorry, the data matrix, the info, and then the condition, the treatment. In my case, it's just the column name of info, right? So it's just going to be this column name, tilde condition. Stop. Sometimes it autofills stuff that I don't want it to autofill. Okay, I load that in. Everything should be happy. Everything should be fine. Yes, no problems there. So DDC or DDS, right? It's going to be a very large file. Has a lot of different information in it. Not super important. Not important what's in it. Now, like, we do have a couple of really lowly expressed genes in here, right? They do exist. Right, in this case, DCNL1 is kind of a lowly expressed gene. And lowly expressed genes are kind of hard to measure for differential expression, right? Because one read, can you really tell a difference between one read or two reads? There's probably not really a difference. So we get rid of the lowly count reads, the lowly expressed genes. So I'm going to assign this variable called keep, and it's basically going to just sum everything across a row. So it's going to make a row sum, right? I'm going to sum all the rows within, sum all the information in a row, make a row sum. Basically, if that row sum is less than 10, get rid of it. Basically, among all of these samples, if there were only 10 reads counted total, get rid of it. We don't want to deal with it. So we're just going to get rid of some lowly expressed genes. And so that's row sums counts. This is just a specific function. I think it's a DESeq function. DDS for that variable. And then I'm going to set it equal to 10. Some people will do equal, bigger than or equal to 10, right? We're just keeping those. Those are the rows that we're going to keep. And then we're going to take this one step further, because if we actually run keep, it's just going to tell us the rows. It's going to be a logical thing. It's just going to tell us the rows where it's going to be a very long matrix or a very long list of genes where that is equal to true, where the row sums is greater than 10. It's just going to be set to equal to true. So we're really not keeping anything. We're just setting that parameter. And then we can actually throw stuff out or keep the rows of interest by doing DDS is equal to DDS matrix keep comma. Again, it's just going to the row names. The row names where this condition is true, we're keeping those. We're only going to keep those and make that our DDS data set. Remove lowly expressed genes because they kind of suck. Okay. So that's kind of the next step, is to get rid of lowly expressed genes. Now we can actually run the main DESeq program. And it's really not that hard to do. It's just DDS. We set a variable, and it's just DESeq, and then DDS, right? The actual matrix name. Okay. So let's see. Make sure everything is run through here. Okay, that's changed, and now just running the main DESeq, right? This is going to do all the calculations that DESeq does to estimate differentially expressed genes, and so it's going to do some normalization, right? Because every library size is a little bit different, and if we have a library, say, that has 1,000 reads in it, and for a specific gene, 100 reads mapped to that specific gene, right? So 100 out of 1,000, like 10% of all reads mapped to a gene. But we have another library that's 1,000,000, that has 1,000,000 reads in it, and say 100 reads mapped to that specific gene. Well, those read counts are different, right? Because we have to normalize for the actual size of the library, the total number of reads that actually mapped to our library. We have to take in account consideration into library size. And so this DESeq function go ahead and does that based on a normalization technique that I unfortunately cannot explain. It's very, very statisticy, don't understand what's happening, but I do know it's part of doing estimating size factors and dispersions, whatever that means. So, nonetheless, we can extract those normalized read counts. So we can do that, and that's not a bad thing to do, because I kind of break down my DESeq stuff into two steps: running the program and exporting the files of interest, and then doing some graphics with those exported files. So I will want the normalized read count from my DESeq stuff. Export normal read count. And so I'll try to demonstrate how the read counts will vary once we normalize and correct for different library sizes, dispersions, and size factors, blah. Let's see how I do that. So I'm just going to make a new variable called norm counts. And so I'm going to use count. Again, this is just a counts function from DESeq. DDSDE for that specific variable, and normalize is equal to true. And let's see how this looks. Error, normalize, unused normal, I probably misspelled something. Normalized. There we go. And so let's see what this looks like. All right, so here's our normalized read count. And so now we've taken into consideration, right, library size. And so these numbers will kind of smooth out a little bit. The row numbers will actually smooth out a little bit, especially if we compare it to the raw data. So if we have this row 1, RNF 14, 108, 114, 108, 94, 28, 124. But they're normalized, it's 111, 93, 108, 82, 36, and 116. So a little bit different. Again, it's just correcting for different library sizes and other things that are happening in the data. We can't. It's stupid to estimate expression on non-normalized data. We always have to correct for variation in the data, and the variation of the data, and that can include size of the library or the number of mapped reads, standard deviations, etc. And again, I want to export this. Actually, I don't want to just save it. I want to actually export it. So I can use write.csv as norm counts. And save it as a specific file. Say normal dot water v 15 psu. Why not? .csv. And hopefully that will go without a problem. Okay. Okay, and so now that's hanging out in my directory. So now I have the water normalized recounts alongside of the actual count data. Again, this is just stuff that I will eventually need later. But again, we can kind of look at that for a second. CSV, and we can actually see the normalized recounts. So once we have it normalized, this is the data that we actually want to work with. If we're going to plot stuff out, we're going to plot out the normalized recounts, not anything else. Okay. All right, and now we can actually look at our DESeq results. So the function to do this little bit of magic is going to be res, or results, sorry, results. And so again, results is just a function of DESeq. So I can do DESDE, the main output of DESeq. And then I'm going to set this alpha is equal to 0.05. And so what this alpha is, it's effectively a corrected p-value. So what I'm saying is, give me the results of the output, and whatever has a corrected p-value of less than 0.05, that's going to be a differentially expressed gene. So I'm just going to save this as res, results DDS alpha 0.05. Okay. That's taking an unexpectedly long amount of time to do. Hopefully nothing is going wrong. There we go. Res done. And so now we can actually look at the summary of our res. We can summarize the output of that result file with summary res. And so summary is specifically a, I think it's an R base function, but we can do summary of res. And so what that tells us, that output tells us it measured expression for 11,000 genes. It measured differential expression for 11,000 genes. And we have this corrected p-value up of 0.05. Right? If this p-value is less than 0.05, we'll I'll discuss this in a minute. And then we have these LFCs. And LFC stands for log fold change. And I'll explain log fold change here in a second, and what that kind of means. But ultimately, we do have differentially expressed genes in this data set. 193 are in an up orientation, or they're upregulated, and 77 are downregulated. But again, we have to understand our context a little bit more to understand these LFCs, or the log fold change. So I'm going to do a couple of things to export this out a little bit better. Because I don't necessarily care about summary arrays. It's nice to give me a summary of the output of DESeq, but I need more details. And so one thing I'm going to do is I'm going to order my output. So if I look at res, I think we can see res. No, we can't see res, not well. I can type res into that, and it tells me column information, what's actually hanging out in res. So we have this base mean, log fold 2 change, a stat, a p-value, and then adjusted p-value. I'll get to all that in a second. I'm just going to order this stuff out to be easier to look at. And I'm going to write that out. Okay. Make sure all those are there. Okay. Okay. All right. So whenever I'm ordering this, whenever I'm ordering this, I'm ordering it based off of the p-adjusted value. Regardless of what the code actually says, what it's doing is I'm reordering my columns, sorting out my adjusted p-value from smallest to largest. So it won't be random. It's much easier for me to see the differentially expressed genes. And then I just wrote that to a CSV file. Again, it's just easier to look at as a CSV file than uploading it into DESeq results. Okay. And so this is a file that I need. I need that file. These two files are what I need. I have the normalized read count, and I have the differentially expressed or not. Those are my most two important files out of this entire thing. And honestly, within 26 lines, less than 26 lines, we've done a differential gene expression analysis. Once you kind of figure out what's happening in the manual, it's pretty easy to do. But let's actually look at the output of DESeq. Water versus PSU. All right. So I have this file. This is my results file. And so I have the gene name in the column, or gene name as a row. And base mean is what is the mean expression among all 6 libraries, right? This is a very highly expressed gene, thousands of reads averaged across all libraries. Then we have this log2 fold change. And this is going to be a positive or a negative value. And it's going to more or less tell us the direction and intensity of the change in expression. I'll get there in a second. And then LFC, SE, and stats, whatever. I think the next important thing is going to be the p-value. And so this p-value is the raw p-value. And so each one of these genes is independently tested for differential expression. And as I understand it, it's a reasonably simple stats test. Not super different from a t-test. And so whenever we do a t-test, we're effectively measuring for a difference in means between treatments. And so a p-value will effectively tell us, in some ways, a probability that these two means are actually different. And they're different by chance. So p-value can be interpreted as these two means are different by chance. And the lower the p-value, the less likely those two means are different by chance. And so, again, we have these two means of the treatment versus water versus 15 PSU. And if this p-value is less than 0.05, right, it's likely different, not by chance, right? There's an explanation. However, a p05 is basically saying, okay, this will happen by chance. These two means will happen by chance, are different by chance 5% of the time. So if we do 100 tests and it's all on kind of random data, 5% of those tests will come back as significant. They're significantly different from each other, says the stats. So 5% will be different by chance. And so what we have to do is, since we've done basically 11,000 tests, and we know 5% of them, at least 5% of them, are going to appear different by chance, we have to correct for that. And we do that by using this adjusted p-value or corrected p-value. There's a handful of ways to do this, but it's effectively kind of like saying just lowering the threshold of significance. So instead of saying like P05 is going to be a significant result, let's say P001 is actually a significant result given how many tests we've actually done. It's not exactly what's happening, but it's kind of what's happening. It's just going to be an adjusted p-value to correct for differences that appear different, although it's just random noise. So we care way more about the adjusted p-value than we do the actual p-value. All right, now this log fold change. Let's talk about this log fold change metric, because this is one of the more important metrics in this output file. So, gotta have a new Excel thing going on, so we can at least kind of see what's happening. So let's say we have our two means. We have our water mean and then our 15 PSU mean. Okay, and so we have these expression means. And so I'm just going to do increments of 5. 10, 15, 20, 25, 30, 35, and 40. I did this earlier today. I think 50 was good. There we go. Okay, so let's say these are our water means, expression of different genes, hypothetical, that vary between 5 to 55. Now I'm going to do this in the opposite direction. Again, we're just going to see what's happening whenever we do a log fold change. So I'm going to start at 55 and go down. Okay, so I'm going to color based off of whether these numbers are higher or lower. I'm going to color code if the number is higher. So in this case, water expression is lower than it is in salt water. So I'm just going to color anything that's lower in blue, just so we, again, not that way. Cells, there we go, light blue. I'll do. These will be light blue because they're going to be lower, and I'm going to color these higher. Again, just because they're ugly, just so it's easier to see. And those are higher. Okay. And so when we're taking a log2 fold change, what we're basically doing is taking a ratio. In this case, it's all going to be the same. So we're going to take the ratio, the water expression ratio, by the 15 PSU, the saltwater ratio. So this is going to equal to that divided by that. Okay, and that's going to be true for all of these things. And so what we're seeing is that we'll see a fraction whenever water is lower than salt water, or we'll see a not an integer, but a positive or a number greater than 1 whenever water is higher, whenever genes in water are higher expressed. And when they're the same, we'll get they're equal to each other. So that's our ratio. And so our log fold change, and we should see what happens when we take a log fold, a log of this ratio. So I'm going to take that ratio and I'm going to set it as a base of 2, log base 2 of this ratio. Okay, negative 3.45. I'm going to do that for everything. And so the point being is that we should see a pattern emerge whenever we look at our log fold change. When water is lowly expressed, our metric, this log fold change, is going to be negative. But when water is higher, because the ratio is a whole number, or at least it's bigger than 1, this will be a positive value, right? So the log fold change becomes positive. And so if we kind of remember what these values are, it's kind of like this is kind of like a pseudo normalization. But whenever we take the log 2 of a number, of a ratio, it tells us the direction and intensity of that change, right? So it's saying that compared to water, water is lower expressed because there's a negative log fold change. Water, the genes here, in these case, water and water are lower. But when the log fold change is positive, it means that the genes expressed in water are higher expressed than the salt water. And so you see that the intensity is the same, right? And so it tells us the direction. Now if we kind of remember, like, understanding like where this is coming from, so this is basically saying 2 to the x is equal to y. But whenever we transform, alternatively, log 2 of y is equal to x. Right? Remember, there's just this relationship between exponents and logs. So basically what we're getting here is an exponent. It's the equivalent of an exponent. 2 to the what describes the change. And if that number is positive, it tells us that, in this case, genes in water are higher expressed. If it's negative, genes in water are lower expressed. Right? I think that's the more important thing. It's like, it just tells us the intensity of the change in the direction. So whenever we look at our log fold file, if we see, so what I know this, the condition is water versus 15 PSU. So water is going to be first. And so for this gene, lipo, this gene is more expressed in water by a little bit, by a log fold change of 1.4. That's a significantly differentially expressed gene, and it's upregulated, so it's more expressed in water samples. But over here we have this LEAP2 gene, and the log fold change is negative. It is significant given the adjusted p-value, and so it's basically saying in the salt water, this gene is more expressed than in regular water. So there's a change. And so we can kind of plot these out. So ultimately, whenever we look at our summary results from our DESeq stuff, whenever we look at our log fold change, when our LFC is above 0, it's saying that in our water samples, 193 genes are more expressed in water than they are in salt water. And by contrast, 77 genes are expressed more in salt water than they are in water. And so we can kind of determine that by looking at the names. So like, I gotta find it in a script. Nope. Oh. I think it's results. Results names. DESeq. And so it's telling us our condition is water versus 15 PSU. So in this scenario, we're following this scenario where water is in the first column, PSU is in the second column, and we divide PSU or water by PSU. Right. So again, negative, we see negative log full change, it's higher expressed in the PSU. If we see a positive full change, it's more expressed in water. And so we can kind of visualize this result using a built-in function to DESeq. That's called plot MA. And so plot MA is basically going to put the gene expression or the mean expression on the X and then the log fold change on the Y, and then color code significant based off the p-value in blue. So that's DDS, DE, and I'm going to set a Y limit. -5 to 5. That's going to take a couple of minutes to finish, or a couple of seconds to finish. I think everything runs a little bit slower while I'm streaming. There we go. And so we get this kind of ugly looking graph, but it tells us a lot of information. So again, the y-axis is just the mean read count or the normalized read count. And then the direction is the fold change. So again, if we see a positive, it's biased for water. If it's negative, it's biased for salt water. And so in most cases, genes are, if they're differentially expressed, more often than not, they're higher in water and they go down in salt water. So it is a result. There is a difference in gene expression, at least for 100 plus some odd genes or 200 genes. There is an expression difference. They are differentially expressed. So that's kind of interesting. We'll take this a few steps further to make some more plots on the next time, and then probably next week we'll figure out what these genes are. We'll figure out how, if there's a pattern to the functions of the differentially expressed genes. So that's going to be kind of the next step after that. I'm going to save this script so I don't have to write all this crap out again. And we have the main output files that we can plot with after this. So I'll see you all again on Friday, and we'll keep plotting this stuff out.